Hello, this is Team Row Ashno Kai, and we are presenting on number 12. I'm going to be doing question 1 and 2. Sorry, it's taking a little bit long to come. There, guys?
Yeah, I'm still here.
Okay, I'm still here too. Good. Ooh, that just scares me that last time. Okay, so number 1, they basically is just a mix and match. They have all these terms and they want to know where they go. Number A, they want to know which is an example of a regulatory protein. And I have repressors, activators, and the CAPE protein, which is an activator protein. And then for part B, which example is a small effector molecule? That's going to be allolactose, which binds to the LAC-I protein, and an inhibitor, an inducer, and cyclic AMP. And C is which of them are sequences found within DNA. And the -10 + 1 and -35 are all sequences part of the LAC promoter, and the operator is a sequence on which the repressor binds, and LAC-Z and LAC-A are structural genes. For D, which of the above is not involved in the LAC operon? Inhibitors are the only one that is not involved. And now an inhibitor, it will bind to an activator protein, but the activator protein that we have in the LAC operon, it has a small effector protein that binds to it for it to be able to bind to the protein. And so it does not have an inhibitor, at least that was mentioned in the lesson, which would bind to that which would be involved in the LAC operon. And structural genes will be LAC-C and LAC-A. And for part 2, they want to know the LAC operon is regulated by 2 inducers. And that's going to be allolactose, which will bind to the repressor regulatory protein, and the cyclic AMP, which binds to the CAP protein, which is an activator protein. And the binding site for sigma factor and RNA polymerase is called promoter, LAC promoter, or LAC-P, while the operator, LAC-O site, is the binding site for the activator that regulates the LAC operon. 2C, the enzyme adenylene cyclase converts ATP into CAMP. And for 2D, the -35, -10, and +1 sequences are found within the LAC promoter portion of the LAC operon. 2E is the LAC gene encodes the LAC repressor, while the LAC-Y gene encodes lactose paramase. F, attenuation, is when gene transcription is terminated prematurely, and in E. coli, a strain that contains 2 copies of LAC operon is called a mirrozygote strain. So you have the chromosome and then also a F' plasmid. All right, now we will hear from Noah on question.
Yeah, question 3, right? Can you all hear me?
Yep, I can hear you.
All right, cool. So I believe I'm screen sharing. Cool. So for number 3, it says, suppose you are examining a lac operon in E. coli, and your experimental activity of this enzyme B-galactosidase is measured by including XGel and IPTG in the growth media. So the IPTG can mimic allolactose, and IPTG can bind to the lac repressor and release the repressor from the operator. So it's just like your LAC-Z, I suppose. And X-gal is a molecule that is a substrate for this B-galactosidase and turns the E. coli cells blue when metabolized by the beta-galactosidase. So in parts A through E, there's these 5 different culture solutions, and it wants us to determine whether the substrate will actually be present, if it will be blue, if the cell is blue. So for A, the IPTG is absent, so that means that the repressor will never be removed because the IPTG mimics allolactose, which is what removes the repressor from the operon. So it'll go through once and then be stuck. So even though the XGel or galactose is there to produce the blue, it's never activated. So B, the IPTG is present, so the repressor can be removed. Glucose is absent, so there's the production happens, and the X-gal is present as well, or that blue substrate. So B will produce blue E. coli cells. For C, IPTG is present and glucose is absent, but the X-gal is absent, or that blue substrate is gone, so there's no way that blue can be produced. So no, you will not have blue for C. For D, the IPTG is present, but the glucose is present. So that means that there's not very much of the CMF being made, so there's not enough energy to have the cycle run. So there wouldn't, while it's still possible because the blue substrate, the X-gal, is present, there will not be enough to cycles to make it blue. So IPTG is present, and glucose is present, and X-gal is absent. So it's just like D. First of all, there's no X-gal, so there's no blue substrate, so it can't be produced. And even if it was present, like in D, because there's glucose, the cycle won't be run enough in order to produce those substrates. Okay.
Okay, so on to question 4, which is my section, my question today. I got the long end of the stick, so it's nice. So pretty much in question 4, it's asking us, it's giving us some genes, and it's telling us that some of these genes are broken, some of them aren't. So going through these in type A, or then it's asking us if it's going to be constitutive, inducible, or non-inducible expression for the LAC-Z gene, which is a little bit further way down, where it's just the one gene down from the LAC-O. And so in type A, it's going to be inducible because we have wild type for all of the genes. And so it's going to work. It's going to be wild type. In part B, we're going to have a non-inducible because the IS, which is the repressor, the LAC-A repressor, is going to be stuck all the time because allolactose can't bind to it and release it from the O site. So it's going to be non-inducible for that one. Number C is going to be constitutive because we have a O site, or a LAC-O site, that the repressor can't bind to it. So it's always going to be producing, pretty much always going to be producing the Z gene. In part D, we have 2 sets of, or I guess part C, no, yeah, part D. We have the IS, which is going to work its way. It's pretty much the releasing of the lac oocyte, and it's going to work, and it's going to bind over to both sides of these chromosomes. It's going to bind to the chromosome as well as the plasmid, and it's going to make everything stop. And so it's going to be non-inducible because the allolactose can't bind to it and release it from the gene. So it's going to be non-inducible. And then in part E, you have the this repressor, which pretty much blocks everything, but you also have a lac oocyte, which the repressor can't bind to. So it's going to be constituted because the plasma is always going to be producing because nothing can bind to it. And so that's my explanation of number 4.
All right, so I also have number 5. And so after much deliberation, this is the conclusion that we've come to. It's a consensus. So hopefully we got this right? So for part 5, we're basically saying, what if the experiment given in this lesson was done with these certain strains of E. coli? In each situation, there will be, for each strain of the E. coli, there will be lactose induced or lactose provided and lactose not provided. And so we'll have to determine how many, through one cycle, how many lactoses are produced, which gives a yellow color. So 0 will mean no lactose, 1 will mean that 1 lactose is produced, and 2 will mean that 2 is produced. So it'll be super yellow. Okay, so over here on, is my camera showing? Okay, cool. So over here on A, we can see that in the genotype, the lac-I is absent, is defective. So that means that the repressor will not be generated, and the cycle will continuously run through and produce lactose. However, the promoter is also defective. So it will get to the promoter site, and because it's defective, it won't start. So even though it would be constitutive because the promoter site is malfunctioning, whether or not there is lactose or not lactose, nothing will be produced. So for part B, you have all wild type, but the O is a defective operator, which means that the repressor site, or the repressor, cannot bind to this operator. So that means that even though this I is positive and the repressor will be produced, it cannot bind to the O site and stop production. So that makes it constitutive. So whether or not lactose is there or not, it will be produced. So that will both be 1. Now for C, the chromosome genotype in E. coli is all wild type. So it follows the basic rules. However, in the plasmid, the O is defective, which, like we just explained, means the repressor cannot bind to the operator site, even though it is generated. So if we have lactose, then the I site will be nulled, the lactose will be generated, removing the repressor. So we will have lactose produced from the chromosome. And because it doesn't matter for the plasmid whether or not the I site produces a repressor or not, it will generate. It will also produce lactose. So there will be, if there's lactose present beforehand, there will be 2 lactose produced, or it'll be super yellow. And in one, if there's an absence of lactose, there will only be 1 produced, because the plasmid will produce no matter what, but the chromosome follows the normal rules. And if there's no lactose, there's no allactose generated, and the repressor will not be removed. Now for D, we see in the chromosome that it is constitutive, that the it will produce no matter what, because there's no repressor that's generated. And the promoter in the plasmid is defective, so nothing will happen. There will be no transcription generated. So no matter what, the plasmid, lactose or no lactose, will do nothing. So we only focus on this D chromosome. So on this D chromosome, or on chromosome in D, because it doesn't matter whether it's lactose or not, because there's no repressor generated, we will have 1 and 1. So it will be yellow. Now in E, we have a similar situation. The chromosome is the same as in D. There's that lac I is negative, so it will be constantly generating. However, the plasmid has this IS, which means that it will bind, the repressor can bind to the operator site, but lactose cannot, which means that the repressor will never be removed. So it may go through the cycle that one time, but then it will be stopped. So whether or not there's lactose, it will be 0 or no yellow. And that is the explanation for number 5.
All right, thank you, Noah. I'd like to thank him also for doing double duty for Ashley when she was supposed to do number 3. That's okay. Just for the grader's convenience, for 3A, it was not blue, 3B, blue, 3C, not blue, 3D, not blue, and 3E, not blue. So the only one that was blue was 3B. All right. Thanks for posting that thing, Roy. That's funny. All right, and we'll see you guys all next week.